This conference will now be recorded.
All right, folks, good morning. So we're going to start the morning off with looking at some interesting examples of a species that's created, but it's not in the case of, say, we're taking animals from different species, right? Same genus. Recall the whole. Let's go here first. Let's look as far as. So this is slide 12, and we're looking at as far as hierarchy is concerned and how we go about grouping the different organisms throughout the animal kingdom and the plant kingdom and such. And so remember that we have eukarya, right, is one. Then we have there, as far as the archaea is the other, as far as domains are concerned. And you'll see here that we have the eukarya. These would be the organisms, again, that have membrane-bound organelles, membranous nucleus, right, with the genetic material within. These are the eukarya domain. And within the eukarya domain, we have kingdom Animalia. And then we go all the way down, and we're looking at here as far as genus and species. So when we identify some type of, whether it be plant, animal, organism, whatever it may be, we're using the genus and remember we call this the specific epithet, also known as the species, right? So we're looking at genus would be Ursus, species would be Americanus. So it's Ursus americanus, right? And so what we're looking at here is that within a species, we can have reproduction take place, and we could know that then they will produce offspring that are also fertile, okay? That can also reproduce after their own kind, okay? But when we come to breeding between two different species, right? Same genus, but different species, this is going to create an issue. And so we end up with the Liger, okay? So the Liger, let's see here. I don't remember it off the top of my head yet. So it's because of the Li being in the first part of the term that we describe it as. So the Liger would be the male lion, female tiger. And these are magnificent animals. But again, this would not be an animal. This is a huge animal. And it would not be able to be viable in the wild, okay? I think even that this is at the place where if you saw oh, what was it, Tiger King or something like that, or that whole wackiness that went on that was on Netflix and such. I think it's been bred. This Liger has been bred in that location, and just one of the locations that you saw there, just strange stuff. Anyhow, Tigon would be a male tiger and a female a lioness, a female lion. Okay, and you can see that there are different presentations of this Tigon. And again, these are not fertile. Okay, so we're producing this hybrid where it's not going to be able to reproduce its own. You'll have to have the two different species coming together to ever create another Tigon. Okay, Tigons themselves, they are infertile, as well as the Liger infertile. And one more here, this is cool, to just see the difference. Looks like a grizzly, and yet it's very, the fur. So this would be a female polar bear, the Pizzly, female polar bear, and a male grizzly coming together. American brown bear, right? The huge bear. So both of these bears, massive animals, and producing this animal called a Pizzly. Okay, so just thought I'd show that to you all there and just see how interesting this is. What I'd like to do right now, we've been talking about the phylogenetic trees. So let's just take a look at examples here. So here's examples of, and it's the same example of the same tree, okay, just in different presentation. But you'll see here that this phylogenetic tree here and here, we're looking at evolutionary ancestry, okay? So we're trying to see that common ancestor, right, at the root here, common ancestry, and then we have these different species, present day, right? These are more in the past, okay? And really, again, common ancestry. We talked about, we mentioned about the tree of life, and told you that, and I mentioned to you that it's very similar to. So here we go. Here's a family tree. So this is the Miller family. let's open this up here. And you can see how with this family tree, and we can all have some type of family tree. We know that we have descendants, and we have ancestors in the past. You know, here we're looking at 1758, 1763, they're born, and then we end up having all of these relatives till we get to the point where those that are living, right, we're here are the living relatives, and we're seeing as far as you know, all the branches of the ancestry that has brought about to you and I, or this in this family, the Miller family, for Kathleen, for Grace, for Vaughn, right? All these relatives here as a result of the heritage here at the root of the family tree, Isaac and Ellen, right? Quite interesting. But we're seeing again here, as far as with these phylogenetic trees, we're looking at evolutionary ancestry, okay? And again, looking at it from that point of view, so we're looking at there's a common ancestor at the root here, and then there are branching events that will take place at these nodes, these areas where there is a branch that'll take place, a divergent branch, creating different species, okay? As a result of different criteria, these characters that we'll talk about in a moment. What I'd like to now bring you back to, before we talk a little bit more about this, and again, be more descriptive in discussing regarding these phylogenetic trees. They're also known as, we can use the term cladogram as another term that we can use, an AKA for these phylogenetic trees, okay? Would be talking about and discussing, all right, so here we're looking at a phylogenetic tree. This was back in 2002. A paper was produced regarding this criminal case that was back in, let's see now, here, let me go to my notes, 1994. So listen to this here, folks. So here we're having a situation where we're able to take the information that we're able to create these trees, right, through research and such, and through the scientific method. We're able to determine if someone was given an illness by a doctor as a result of malpractice, and really a very unethical thing that was done and performed. Let me explain it to you here. So in this case, there was a gentleman who was a gastroenterologist who had a fight with a girlfriend, and so there was an issue. They were angry at each other. He was angry at her, and so what he did was he took the blood from one of his patients who was HIV positive, right, had the virus, and told her he was giving her a vitamin injection, like a B12 injection for give you energy and such, and really it was an injection of blood material from this HIV positive patient. So he gave her HIV, right, the virus, by injecting it into her. Right? And that's evil. That is really, yes, yeah, you should, Brian, you're right. Oh my, because that's evil. That is wrong. He ended up getting convicted. And here's how one of the parts of the investigation led to his conviction was that what they did was they were able to determine that this was the hypothesis. They were thinking, well, he must have done this. He must have given this to her. And his defense for the gastroenterologist said, well, hey, she could have got it from anybody in the area, not just from, you know, it didn't. He didn't admit to doing this, right? So he's going to say that, sorry, that his defense said, no, you know, she could have she probably more than likely gotten it from sleeping around or whatever. So here's the deal. What they did was they took the genetic sequences of the virus from the patient, the person who they believe was the HIV positive patient that came in to the doctor's office within the same day, I believe, if I'm not mistaken, the same day that she was given the B12 injection and given HIV virus, right? So they took his information, they took his blood work, did sequencing, genetic sequencing of the virus, the specific HIV virus that he had. They did genetic sequencing of the virus that the the person who was the victim, right? So here's the victim, here's the patient, the doctor's patient, here's the victim, the ex-girlfriend who was given the virus. And right over here, you'll see all of these would be examples of HIV viruses that were present, the different strains of viruses that are present within, and the sequencing that was done on people who are in the community where this woman lived, okay? And what they found was that by doing the sequencing and looking at and creating this phylogenetic tree, they saw that. Now look at this for a moment here. Do you recall that at these nodes right here, right here, 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 and here, right? Where we have right here, no, right here, sorry, right here. And you'll see these nodes, let me show you here. So these nodes, look at this image right here, you can see these nodes. These areas, we would say that these are areas of common ancestry, okay? Common ancestry. So let's go back then to this image here. So they were able to determine through, again, the genetic sequencing of the virus, of the patient, of the victim, that they were more closely related right here. Here's the patient, here's the victim, right here, than this victim was to the other people within the community that had HIV virus in their sequencing. It's quite amazing. This led to his conviction, and this is back in 1994. In 2002, a paper was done regarding this study. So quite interesting, folks. How we can see how it's not just a matter of just, okay, we're learning how to create these trees and what the different terms are for these trees and such, and how to read them. And again, I'll be reviewing that today with you all. But see how in the real world, how this has made a difference as far as in the conviction of a criminal. And truly, he's a criminal. And he has taken his oath as a doctor and just like stepped on it and done evil. And that's totally unethical, totally wrong. And he deserves to be in prison, and he is. So, all right. So interesting, right? How we can see how these phylogenetic trees, how they can play an important part in what's going on in the real world, and how, again, it led to this conviction. So I thought you'd find that very fascinating and interesting. Let's go to, all right, so we looked at this hierarchy as far as the taxonomy is concerned. Now let's go here, and then we'll go to the next slide. Again, we're just trying to review and go over some things, and I want to make things more relevant to you all, and also help you to understand it a little bit better here. You'll see here, as far as the phylogeny, this evolutionary history, that's what these trees that we're creating are trying to show, the evolutionary history. And it's really the hypothesized evolutionary history, because we weren't there, were we? So we only can make an educated thought process here as far as the hypothesis is concerned, in order to determine what the evolutionary history of the organisms are. And really knowing that the root of the tree is the common ancestor for all the species. So as we look at this image here, here's the root, here's the common ancestry. Here we have to all of these species, right? All of these species. Now let's go to, so these in 16 and 17, it talks about these nodes, these areas where we have the branching, the divergent branching, creating separate species, okay, in the evolutionary history, okay. We see that is the term the clade. I said to you that we could also call the phylogenetic tree a cladogram, okay, cladogram. You should know that term, realize that can be used interchangeably, right? We're going to see things like sister clades and sister species and such, and this deals with relationship to different organisms. So let's do this here, and let's take a look at this image here and see that as we're looking at this image, okay, and we're not really seeing any specifics, we're just seeing here, all right, this is representative of species A, species B, C, D, E, and F. Okay? Well, a clade this would be a group, a closely related group. Okay, and it has a common ancestry. So we look at these nodes right here, these areas where there's a branching that takes place. So we have a node here, 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 and here. Okay, and so what we're looking at, folks, is that these are areas, places of time. Remember, this is a timeline here. We have the present day here, present day species living in this world today. This is here. This is the present. Here's the past, and then we're going. This is a period of time. This could be, so it represents evolutionary change. So this is millions of years that they're talking about. Okay, evolutionary change over millions of years. Okay, so but we're not giving you specific time periods. We're just saying that, you know, here we would have this. Let's hear this term, common ancestor, as far as most recent common ancestor. So in the case of species A and B, here's the most recent common ancestor, this node right here. Okay, this node right here. Now, how about this node right here? Well, this node right here would be a common ancestry of all of these, A through E. Do you see how they all come to this node right here? They all do. But how about F? F does not, right? So that's not part of its common ancestry. But this node right here, common ancestry for A through E as far as these species are concerned. So a clade is just a grouping, right? A grouping of these that have a common ancestry. So C, D, and E, this would be a clade because here's the common ancestry for, right here at this node, for the three of these species. Again, for A and B, here would be the node. That would be the common ancestor for A and B. Just terms that we use, these terms clade and such. You'll see here as far as, let's minimize this, and then look at, so this one gives you a little bit more as far as specific names and terms, and also as far as characteristics are concerned. So this helps you to see that. So what takes place at this event right here at this node? So here we have the root, and know that this is the same as this. They're both identical. They're just given in a different presentation so that they can do these phylogenetic trees in different patterns, in different ways, yet still this is identical to this, okay? Right? So you'll see here that we have this first node, and this is where there's going to be an event that takes place. We'll have a branching off as far as the ancestry is concerned from vertebrates to those that molt their exoskeleton. Okay, so in the case of lobsters, spiders, right? Then we have here, we come up to this next node or this next event that occurs, and we'll see here as far as those species that have lungs and those that have spiny rayed fins. Here, here, and then for the lungs, we'll have, so these all have this characteristic, this character trait of lungs. But here we have an amniotic egg for these lizards, birds, mammals, whereas lungfish, no, that does not reproduce, that does not have an amniotic egg. And then we'll move here as far as to see UV light, lizards and birds, mammals do not, right? As far as being able to see it in the spectrum, that's different than. So you'll see here, folks, that we have, again, in a different pattern, but still the same, identical. And we're seeing here that at these different nodes, at this common ancestry, then there's going to be an evolutionary event that takes place to create two specific differences as far as the different species. And then these in turn will continually have an evolutionary event occur where we'll have change take place as far as the characters are concerned until we get to the point where here are these species that are alive in today, in the present, okay? Again, common ancestry, okay? And as we looked at, and I showed you, so here as far as in the evolutionary tree is concerned, there we go. All right, so as far as the tree of life is concerned, as far as evolutionary history is concerned, right? So we would say is that really at the very foundational, here we go. So at this at the very foundation here, right? At the very root of the tree of life for all living creatures within the world today in the evolutionary aspect of this phylogeny, we would say that it would be a single-celled organism, a protist, which in turn, all of these organisms would result as a result of evolutionary changes. You'll see here that it shows you, here's the root, the branches, here's the nodes. At the nodes are where there's a change that will take place, and there'll be a divergence, one trait to another trait, creating separate individual species. Okay, so let's get at that, and let's go to, I'm going to go back to the PowerPoint here, and I want to show you some terms regarding classifications. Okay, so this term monophylactic, polyphylactic, and paraphylactic. And here you're going to see an example of the images here in a moment. But these terms, you should have an understanding of what's going on as far as the differences between these terminology and how we describe it on the phylogenetic tree. So when we think of monophylactic, we think of common ancestry and common ancestor and all the ancestors that are a part of. So all, that term all would be all inclusive. So an ancestor and all of its descendants, okay? This would represent this monophylactic taxon or group, okay? Polyphylactic, we're looking at here, phyletic, would be including the species from different evolutionary lineages. So look at this here for a moment. So yes, they do have a common ancestor here, right, for these, but really their most recent common ancestor would, for this group, would be here for this one, for these two species, and for these two species, it would be here. So do you see how the difference between the monophylactic, phyletic, right here, and the polyphyletic, right here, as far as they're not grouping them as far as common ancestry is concerned, okay? Most recent common ancestry. They don't share the most recent. Whereas a paraphyletic, right, some, but not all. You'll see here that these species, these descendants are not included in this paraphyletic taxon. It's just, if they were to describe and use these terminology, this is what you need to just remember and know. You'll see here that here would be common ancestry at the root, and you're not having all of the descendants, only some of the descendants from that common ancestry. And know this here, folks. So let's I'll show you another something that you should know here. And this would be, as far as in looking at these, right? So I said that they're both identical. So both of these phylogenetic trees are identical. Know that if we spin them on their nodes, if we twist, if we change them a bit on the node themselves and just turn them, it's still the same phylogenetic tree, still the same information. You've just moved things around. So in other words, when I mentioned to you the term sister, right? Sister nodes and such, and sorry, let me just get the proper term here again. Go back to the PowerPoint. Yeah, the sister species. There you go. So the sister species. So know that. Oh no, here we go. This one. So lobster and a spider would be a sister species. They're closely related because here's their most common ancestor in this phylogenetic tree. The perch and the flounder, sister species, because they are having their most recent common ancestor, right, is right there. It's very close to them, okay? So lizard, birds, most recent common ancestor right here. So sister species, understood? And again, you could look over here and see sister species. Here. Now know that as we're looking at this, the sister species just means that they're more closely related because they're closer as far as their common ancestry. Okay? In comparison to, would the lobster and the mammals be a sister species? No. Yes, they are related, very far down, very many millions of years according to evolution, right? As far as the evolutionary tree of life is concerned, right? That there is a common ancestry, but it's pretty far, it's pretty deep in comparison to here as far as these species in the present that live within the present. Okay, so really looking at these types of phylogenetic trees really help us to see as far as common ancestry and also relatedness as far as evolutionary change is concerned and such. See if I can show you. So I want to show you here. Okay, so here, this is showing you, I'm going to increase it in size, and I will share these all with you folks, okay, later on when I get back for my receiving the vaccine there. So what you'll see here is these are terms that I would like you to know. Analogous, homologous, okay? Homologous structures, all these organisms come from a common ancestor, okay? And so they are similar in their anatomical structures, okay? Similar. They have, anatomically, they're similar. So you'll see here that for the human, right, you'll see here as far as the radius, there's the thumb side, number one finger, the thumb, so that would be radius, the carpal bones, and the phalanges, the digits, the fingers. Now look at the cat, look at the whale, look at the bat. We have here, as far as the radius in orange, the radius, the carpal bones, carpal bone. And then we have here the phalanges, the fingers, right? We have here these digits. Do you see how they're similar in structure anatomically? Analogous, homologous, sorry, homologous. They're similar in structure. Anatomically, they have the same parts, common ancestry. Okay? When we look at analogous, different ancestry, okay? Similar function, but different anatomically. Do you see how these are similar, right? But as far as the function they do, yeah, that function can be really very close to what all three of them do with these fin, wing, flipper, right? But they're not, they have different ancestors and they're different anatomically, okay? In comparison to seeing here, as far as homologous structures, there is this sameness within the structures. They're just different sizes, but still, and in different looking as far as the appearance, but yet they're still, again, homologous to the different types of parts, right? Anatomically, structurally, they are similar. And so that would be homologous and a difference to analogous. So really see here as far as the structurally, again, and anatomically, they're different, okay? Yet they perform function-wise, they do very similar type action. Structurally, similar. Functionally, similar. Analogous, homologous. Let's take a look as far as in your PowerPoint here. So we looked at, last class we were looking at as far as the homologous bones here and looking at the stapes, right? And as far as the hymandibula, here's another image here. This is 28. So 28, you're looking at a bird and a bat. And you're seeing here as far as homologous wise, right? So we have here the radius in the ulna, as far as the bird is concerned. The radius in the ulna is fused in the bat, but still present structurally, right? Structurally, anatomically present, okay? Homologous. Yeah, so here we go. So homologous structures are similar in anatomic detail, structurally, right? And in their relationship to surrounding structures. So we saw that there as far as that image is concerned. So I wanted to help you regarding homologous, analogous. And again, I will post the additional images that I'm sharing with you today in documents and resources, okay? The next area of the PowerPoint that I would like to go over, and then, like I said, I will finish up the PowerPoint next week, okay? Is I'm moving to this term outgroup and in-group. So outgroup and in-group, in the absence of actual fossil record to support heredity, okay, and really the ancestry and such, we use what's called outgroup in-group, okay? So in the absence of fossil evidence, they'll use outgroup comparison to identify ancestral and derived characters, right? So those characters, those characteristics, okay? So characters in a clade, in a group, right, are compared to those in related species that are not members of the in-group, called the outgroup. Character states observed in the outgroup are considered ancestral, whereas the in-group would be considered derived. So here's what we have going on. So here's the ancestry here. Here's the common ancestor, right? And so this would be, again, very far down the road as far as in the past, right? Here, the caddisfly would be considered in the outgroup. It does not contain the same characteristics that this clade right here does as far as in this in-group. So in-group, outgroup. They have the common ancestry, but other than that, the other characteristics are not shared by the outgroup. The caddisfly does not have 6 walking legs as far as the but as far as the butterfly species are concerned, 4 walking legs for the monarch and for the yellow palm dart butterfly, right? It doesn't have that. It will have 6. It's different. This is the outgroup. This would be the in-group. That's the clade. That's the grouping. These are more of a common ancestry, closer, more recent common ancestry here at the node. And then there's another term that I would like for you to look at. We'll come back to this in just a moment as far as this constructing of a phylogenetic tree, because we need you to have an understanding of, well, if I take data, how can I create this tree? And what you're doing is you're seeing here as far as, you'll see here that we have the different organisms, right, the different species, and we're looking at the characteristics, the character traits, okay, that develop over time. And let's just go here, we'll go right to the very end. And so we've taken that data, that information, and you'll see here that the lanceolets are the outgroup. They are the outgroup of this whole phylogenetic tree. The in-group would be from lampreys to birds, because they all have, right, common characteristics that the lancelet does not. That would be considered that outgroup. So you see here as far as vertebra. So all of these have a vertebra, have a spine, a vertebral spine, and the lancelet does not. The lamprey does not have a jaw, but all of these groups from sharks to birds have jaws. Keep on moving up, right? And so we're looking at, and we're seeing here as far as swim bladder or lungs, okay? So the shark does not, but all of these at this node, that branching, that event took place, that speciation event took place, where we have now, we've created sharks and now a new species, and this new species will then be the parent species for a descendant of bony fishes, amphibians, mammals, lizards, crocodiles, and birds. Do you see that there, folks? So we've taken this information from this table, and we've been able to create this tree. And again, using that term, that outgroup, in-group, and such. We're going to talk a little bit more as far as how we take examples and create also. I'll do like a step-by-step with you next week, okay, and in class on Monday. I want to hit one more aspect of this information, and we'll be done for today. And it deals with the term parsimony. Parsimony. So the principle of parsimony states that the simplest plausible explanation of any phenomenon is the best. And when we're thinking in terms of creating these phylogenetic trees, the evolutionary history of organisms, right? So the best cladogram, the best phylogenetic tree is the one that hypothesizes the smallest number of evolutionary changes, right? So these changes that are taking place, meaning the character traits, the nodes, the speciation that's taking place here, the smallest number of those changes are taking place here. That would be the more accurate phylogenetic tree in comparison to one that showed more of these events. Let's show you here as far as, so looking at B and looking at C, okay? So we all have the same species, same number of species, right? There's six species. This is present day. This is so right here at the root, This would be back millions of years as far as when the ancestors, the initial common ancestor was. Again, we're not putting a specific date or time, but we're saying though that there would be time represented from here, this node, common ancestor, to these species that are alive and present today in the world, okay? But you'll see here as far as the hypothesizing 5 evolutionary changes or 10 evolutionary changes. So we would say as far as using the principle of parsimony when trying to create the most accurate evolutionary tree for these species, we would say that this one would be more accurate because it's still showing, now if you look at here, right, still the same characters, but they're just done in such a way, it's just created in such a way that it's showing you the least amount of evolutionary changes, and that's what they would say would be more accurate using this principle of parsimony, okay? If someone is parsimonious, they would be very tight with their money and use their money in such a way that they wouldn't be wasteful. So in this case, we're trying to figure out as far as what would be the most we would believe that B would be more accurate as far as when we think about changes taking place in nature than C. Okay, so. All right, folks. So let's see here if I'm missing any other images that I'd like to share with you all. Okay, so again, knowing terminology and such will help us as far as next week there when I give you some examples of how you would be asked questions regarding phylogenetic trees and such. And creating them and such. That's what we'll start off the class with, and then we'll move on into chapter 5. Okay. So what I focus on, what I'm lecturing on and studying, as far as in the lecture periods that we have, that's what you're going to focus on as far as for study purposes. Okay.